2020 జేఈఈ మెయిన్ ఫలితాలలో ఎన్ఎస్ఆర్ ఇంపల్స్ విద్యార్థుల ప్రభంజనం. ఆల్ ఇండియా అన్ని కేటగిరీస్లు 18, 35, 251, 253, 582, 606, 778, 780, 812, 821, 1031, 1039, 1303, 1575, 1654, 1753, 1754, 1978 ర్యాంకులే కాక మరెన్నో ఉత్తమ ఫలితాలు సాధించిన విద్యా సంస్థ ఎన్ఎస్ఆర్ ఇంపల్స్.
Hello students, now we are in the discussion about the biotechnology. For the last class we studied about the introduction, then such a great effort of the scientist Herbert Breuer. What he has been discovered? He discovered wonderful discovery of restriction endonuclease enzyme, or we call restriction enzyme. Itself they are called molecular scissors. Such a wonderful discovery has made by Herbert Breuer. And what was the discovery? He discovered what are called restriction enzymes. So this is most wonderful discovery has made by Herbert Breuer. Right? Now later if you look at another scientist, if you look at, sir, one second. Yeah. So if you look at, yes, sorry, let us continue further. So Herbert Breuer, which has studied restriction enzymes. Now the work of Herbert Breuer later was coupled with another scientist, Stanley Cohen. It was being coupled with another scientist, Stanley Cohen. Now Stanley Cohen is another scientist. Now he also has made wonderful discovery in biotechnology. Now what he has done is he also has taken the prokaryotic cell. What is a prokaryote? He has taken, it's nothing but bacteria, and done experiments, many experiments by taking E. coli. Why he has chosen E. coli? Because you all know very well, in the first year you have studied Escherichia coli is a common inhabitant of human intestine. So it's easy to grow E. coli in the culture medium. If you can provide nutrient media and provide favorable conditions, yes, E. coli can easily multiply and forms million number of bacteria. So Stanley Cohen has done the experiment on the E. coli organism. Now he could able to see that. So what you find within the bacteria? Within the bacteria you know that we find nucleoid and also we find plasmids. So when with the help of advanced technologies in the life sciences, with the development of electron microscope and ultra microscope and nano microscope, he could able to see that the plasmids are freely moving here and there. The plasmids are present freely within the cytoplasm. So he worked on, he has done experiment on the plasmids. What are these plasmids here? The plasmids, we all know it very well. They are nothing but small circular extra chromosomal DNA. What we call small circular extra chromosomal DNA itself is called as plasmids. So here what he observed with the help of ultra microscope, these plasmids, they exist as small ringlets and they are freely present in cytoplasm. They are freely present in cytoplasm. Okay, so we have done the experiment. So if you look at in the experiment, these are nothing but the plasmids. Plasmids are nothing but a small circular extra chromosomal DNA. So you can see the small circular extrachromal DNA. What you find within the bacteria, they are generally called as what? Plasmids. Now he has done number of experiments on the plasmids, especially taking the experimental organism E. coli. So you all know this is small circular extrachromosomal DNA. Now the plasmids which he recognized it as, these are nothing but small ringlets freely present in the cytoplasm. Freely means not as if like free, freely meaning they could able to easily make a move in all directions of the cytoplasm. These are not bonded anywhere. They have access to move, they start moving in different directions. That means they are not stagnant, not stable at one place. He observed that they are keep moving within the cytoplasm from one place to other place. That means here they are freely moving within the cytoplasm, they freely present within the cytoplasm. When he observed as they keep moving here and there very freely in the cytoplasm, now it is observed that it can be easily, and when bacteria cells cultures are taken, he observed that these plasmids are coming out of the cell and going into another bacteria. So he observed that this is possible to extract the plasmid, because he observed that these plasmids are coming out from the bacteria and moving into another bacteria. They are very freely moving. So what he has done, he isolated and developed wonderful method for, he has isolated, so he isolated a small ringlets of plasmids. So he developed the method for the isolation of plasmid. Stanley Cohen isolated the plasmid, small ringlets of plasmids. That became wonderful, wonderful effort for biotechnology, for further study of genetic engineering. So what did he observe basically here? Now when you have taken a culture, so this is, let us imagine this is Petri plate. This is called as culture media. Generally small glass Petri plates will be there in the lab. When you go to higher classes, you can see this Petri plate or glass small with the glass lid, it will be there. It is called Petri plate. In this we add culture media. Culture media contains what? Nutrient. So culture media, it contains nutrients. When we add nutrients, when you drop the E. coli into here, so you know E. coli or bacillus, rod shaped bacteria. So if you look at this, now using the nutrients provided to the Petri plate or culture media, if maintain favorable environmental conditions like temperature, moisture, pH and everything, bacteria multiply for every 20 minutes. One bacteria will become 2, 2 will become 4, 8, 16, like that. Millions of bacteria are produced within very short span of time. Now when bacteria are cultured and when he observed, Stanley Cohen observed under electron microscope, it was found that the plasmid is coming out from the bacteria and entering into another bacteria. The plasmids has the ability to keep moving from one bacteria to other bacteria. Then immediately he removed the plasmids from the bacteria and removed plasmids, removed small ringlets, which we call them what? Plasmids. He developed the method of isolating these plasmids from the culture media, and he observed they act as a carriers. They act as carriers. They act as carriers. Meaning they can transfer something from one bacteria to other bacteria. So he found that they are moving from one bacteria to other bacteria. Simply he called them as carriers. It does not mean that he don't know that they are carrying something. No, they are not carrying anything. He simply found that these plasmids are moving from one bacteria to other bacteria. So simply he called them as what? Carriers. So he isolated these plasmids out of bacteria and cultured them in a separate culture media. That is excellent work has been done by Stanley Cohen. Later in one of the international conference, the Herbert Breuer was coordinated with Stanley Cohen. In international conference, you know it very well, generally it is a gathering of all the scientists across the world, and they will discuss about the work in the related field, in the same field. If two scientists will come, oh I have done this, so this is possible, so I have isolated and discovered restriction enzymes. When Herbert Breuer shared his discoveries with Stanley Cohen, then later both of them worked together in their coordination. So when Herbert Breuer met with Stanley Cohen, Cohen in one of the international conference. Both of them discussed about their work. Later together they coordinated the work and worked together and have developed the technology called as recombinant DNA technology. Now let us see that what they have discovered. So if you look at Stanley Cohen, now he is the one from the bacteria, he could able to isolate the plasmid, isolated plasmids. That means now the plasmid is outside the cell. This is bacteria. And what they have done experiment on which bacteria? E. coli. Right? And here what is the discovery of Herbert Breuer? He developed restriction enzyme. So from the cell he has taken the DNA out, from the nucleus he could able to take the DNA out, and with the help of restriction enzyme he made the DNA into a fragment. So here you have now a number of fragments in the test tube. So in the test tube he has number of DNA fragments. So fragmentation of DNA was done with the help of a restriction enzymes. I hope this part is very clear. What was the discovery of Herbert Breuer? He introduced and discovered restriction enzymes. Such a wonderful discovery. What is the restriction enzymes function? They are also called molecular scissors. They make the cutting of DNA into number of fragments. That means restriction enzymes are involved in fragmentation of DNA. Successfully DNA can be taken out of the nucleus and it can be made into number of fragments by the help of restriction enzymes or restriction endonuclease. This is also called restriction endonuclease. Then he has DNA fragments. Stanley Cohen has plasmids. Plasmids, it is observed they keep moving from one bacteria to other bacteria. So Stanley Cohen isolated plasmid outside the cell. He has taken out from the cell. From inside the cell he isolated the plasmid into out of the cell. Then both of them they discussed together and they coordinated and they have developed wonderful, they have given practical view to the idea what they have got in their mind. Now Herbert Breuer has the DNA fragments. Now together they worked in coordination. Now Herbert Breuer, he tried to take the DNA fragment. Okay, so he tried to take the DNA fragment and it was identified as it got integrated into plasmid. With the series of experiments, with so many experiments, don't ever think wonderful miracles will not happen with one step of experiment. Even though they got the idea that the plasmid can take the fragment or not, they don't know. So they have done series of experiments. In the beginning they failed to do that because integration of DNA into plasmid is not a small task. They have done it number of experiments. Initially they failed, but later after doing so many series of experiments, making changes in their solution, making changes in their enzymes, what they are adding into the solution, after doing so many changes in doing the experiment, after so many series of experiments, successfully they were succeeded in integrating the DNA fragment into plasmid. So Herbert Breuer added the DNA into plasmid and they found that this plasmid act as a carrier, and this plasmid they introduced into another bacteria. Let us take, this is the host bacteria. This is host bacteria. Now it also has a plasmid, but still the plasmid which joined with the fragment of DNA, it was introduced into another bacteria. So now this bacteria successfully received the plasmid which already have the foreign DNA. So the DNA what is taken here? This DNA is called as what? Foreign DNA. This DNA is called as what? Foreign DNA. Why it is called foreign DNA? It does not belong to this organism. It does not belong to host organism. Host organism has its own nuclei, its own plasmids, right? But it is receiving some DNA from foreign organism, some different organism, totally different organism. These genes are not matched with the genes. So that's why the DNA which is taken from some other organism is called as foreign DNA. Such a wonderful discovery has made the step to develop what is called genetic engineering, and successfully they transferred a fragment of DNA from one organism to another organism by using plasmid as a carrier. Later they called this plasmid as vector. Now who is acting as a vector? Plasmid. Plasmid is acting as a carrier or acting as a vector. Now this is used to transfer of, it is used to transfer of foreign DNA, means the DNA which is collected from some other organism, foreign DNA into host organism. And this has made wonderful discovery in developing genetic engineering. This has led to the development of a very wonderful important method of technology which has made called a genetic engineering. Then they finally achieved such a great wonderful effort. Then they have come to the conclusion, yes, it is possible to transfer the fragment of DNA from one organism to another organism. We can transfer some part of genetic material which is called foreign DNA, which is collected from some organism, that can be transferred into some other organism. Transfer of foreign DNA from one organism to other organism, that itself developed into a branch called as, such a great method called as genetic engineering. So it became wonderful step. Now today we can introduce desirable gene. Suppose my industry, if I want to produce the protein, if I want to produce the antibiotic, what I can do? I can take the DNA which can produce antibiotic. I can take outside the organism. I can take it out from the cell, and let us keep it into the bacteria, because bacteria multiplies at a faster rate. I kept the DNA which produces antibiotic, kept in the plasmid. It is used as a vector, transfer into the bacteria, so that this bacteria multiplies in short time. I can produce millions of bacteria, and each bacteria get wonderful ability to produce antibiotic, because antibiotic producing gene got incorporated into the bacteria, incorporated into the host organism. Host organism earlier don't have this feature. Now added with the genetic material, it's a new genetic material, foreign DNA from some other organism it's collected. Now this has wonderful ability to produce the industrial products. Okay, such a great discovery. Now the DNA, when it is added into the plasmid, now they call this DNA as rDNA. What is meant by rDNA? It's called recombinant DNA. What it is called as? Recombinant DNA. What is meant by recombinant? This particular DNA which is collected, foreign DNA from some other organism, once again recombined with another host DNA or another genetic material of host organism. So this genetic material entirely different, recombined with a host DNA, that's why it's called as recombinant DNA, and the technology is called as recombinant DNA technology. It's called as recombinant DNA technology. So people have understood such a wonderful method called recombinant DNA technology. This plays a vital role in the transfer of genetic material from one organism to another organism. That itself is called as genetic engineering. Genetic engineering is a method which is basis for development of the emerging branch called as a biotechnology. So this introduction part, a discovery about the scientist is very important. So you need to remember about the photo of the two great scientists, Herbert Breuer, as well as you have to remember about Stanley Cohen. Okay, I'll just let you know. I'll try to go slowly because this discovery is very important, and here try to understand each and every step very clearly, then only you can understand the lesson properly. Okay, so here I have not taught you anything tough here, it's very simple. Let us discuss once again. So try to listen with full concentration. What is the effort of Herbert Breuer? Because this is an introduction, if you can understand the same concept, we will discuss once again in detail in the lesson. When we enter the lesson, each and every step, how it is possible to take the plasmid out of the cell, how to reinsert them into some other organism. Simply I cannot do with the hand, right? Because genetic material is invisible. First of all, visualizing cell itself is a difficult task. And visualizing nucleus is very difficult. To see chromosome is difficult. In that genetic material is different, and DNA, taking out DNA, it's a very difficult task, right? It's not so easy, right? Simply I cannot take the plasmid out with my cell and I cannot put it back into bacteria, right? It's not possible with the hand. It needs lot of advanced technologies and lot of research work, many experiments are needed, then only people were successful. Today world knows how to transfer the gene from one organism to other organism. That's what we call genetic engineering. Okay, for this introduction is very important. It started with the great effort of Herbert Breuer, who discovered what is called restriction enzymes. These are also called molecular scissors, and the enzyme what he discovered itself is called as restriction endonuclease. Why it is called endonuclease? Within the nucleus, endo means inside, within the nucleus it's responsible for cutting of DNA at a specific site, at specific location. So he has collected DNA from some organism, he has taken isolated DNA out, kept in the test tube, and added restriction enzyme that has made the DNA into fragments. All fragments are collected within the test tube. The restriction enzyme cutting the DNA into fragments is called restriction enzyme digestion, that we will discuss in detail later, right? And when all DNA fragments are there, this is what the great effort of Herbert Breuer. Later when he met Stanley Cohen and he understood about what was the discoveries done by Stanley Cohen, and that's another important great discovery by Cohen. What he observed is, he has taken the culture containing E. coli, it's a bacteria prokaryote, and he found that plasmids are keep moving freely in the cytoplasm. These plasmids are nothing but small circular extra chromosomal DNA, they are ringlets, and Cohen identified they are freely moving here and there in the cytoplasm. Then when they keep moving and also jumping from one bacteria to other bacteria, Cohen isolated the plasmid and he identified the plasmid has wonderful ability to move from one bacteria to other bacteria. So he isolated plasmid out of the bacteria, and this plasmid extraction was the great effort of the Cohen. And when both discussed and combined together, they have done the coordination work. Now the plasmids are ready to move from one bacteria to other bacteria. Herbert Breuer has the fragment of DNA, a fraction of the genetic material. Now they inculcated or integrated the one fragment of DNA into plasmid. Then plasmid is acting as a carrier and vector. When plasmid successfully taken the fragment of DNA, the plasmid is allowed to enter into another bacteria or another organism. The organism acts as a host organism. Today it can be applied to plant cell, it can be applied to animal cell. Successfully we can transfer the fragment of DNA from plant to plant, plant to animal, right? Yeah. So our animal to animal, we can transfer the gene from one organism to other organism. So this plasmid entered inside the bacteria, it start undergoing replication and produced the product whatever is being designed. This DNA what is transferred here, it's foreign DNA, that means it is collected from some other organism, introduced into another organism, it's called host organism. As this is going to recombine with another genetic material, that is why this DNA is called alien DNA, or itself is called foreign DNA, or itself is called as, after combining and entering into host organism, this is called recombinant DNA. I hope here this part is understood. Once the foreign DNA fragment collected from some other organism, it is called foreign DNA or alien DNA. This DNA is new and entirely different from the host. So, a fragment is when added to the plasmid, it is called alien DNA or foreign DNA, means it is totally a different organism DNA. But after plasmid enters inside the host and this DNA is going to combine with the genetic material of the host organism, at that time it is called recombinant DNA, because it is going to recombine with genetic material of the host organism and going to express its own characteristics. That is why it is said to be as what? Recombinant DNA, means some alien DNA, foreign DNA recombining with some other DNA of the host. That is why it is said to be as what? Recombinant DNA. And this technology they named it as recombinant DNA technology. That is nothing but today what we operate called genetic engineering. Right. Now, development of the genetic engineering, in what way it was useful here? Nowadays if you look at the applications of biotechnology, for example, if you look at biotechnology applications, with the development of recombinant DNA technology in the genetic engineering, where they are useful a lot here in development of in vitro fertilization. What is in vitro fertilization? It is nothing but what? A test tube baby. Now this discovery of recombinant DNA technology in genetic engineering has made into application of this into different applications, has involved this genetic engineering was used in different applications of biotechnology. One of the best application of genetic engineering is in vitro fertilization. Till now we don't have any source of test tube baby, but first time they thought that why not, when we can have technology to transfer the gene, why can't we do the fertilization in the test tube. In the test tube they have collected the sperm, they collected the egg and they allowed to get it fertilized, and that lead to development of zygote, embryo, and finally they could able to produce the baby. So you know today it's most familiar one, what we call test tube baby, is one of the applications of genetic engineering. For that, what is the basis? Recombinant DNA technology. Right. Apart from this, there are so many applications like what you call gene therapy. Today we have applications like gene therapy, and even we have technologies like correcting defective gene, right. So this all is nothing but what is the main method applied here? Genetic engineering. In genetic engineering, what is playing an important role here? Recombinant DNA technology. What is playing an important role here? Recombinant DNA technology. In genetic engineering has so many applications, correcting the defective gene, gene therapy, and also like production of antibiotics, nothing but medicines, proteins, enzymes, right? These all are nothing but we get produced at the place called as industries, right? There are what not, there are so many applications today, right? You know, test tube baby and even stem cell technology. I think you know well about nowadays everywhere in every hospital they are creating awareness on stem cell technology, right? So in vitro fertilization test tube baby is one of the most familiar one. What is stem cell technology? Nowadays in hospitals after delivery when young infant is born, so from that they are collecting stem cells, they are preserving in the lab. In future if and all any damage to the body, any accident if the baby met, then the replacement of the cells or the damaged parts can be done with the help of stem cells. So nowadays they are creating awareness on stem cell technology. What not? So here correcting the defective genes, suppose sometimes in case of hereditary diseases, when young baby developing in the mother, so like if the baby is having some defective genes, that may lead to some physically handicapped condition, right? So what we are doing, we are cutting and removing the gene, we are correcting at the time of embryo formation itself, right? So that's nothing but correcting defective gene therapy, right? At the same time biotechnology has wide applications in place called industries, what not, production of antibiotics, even in fermentation we have production of alcohols, right? Food industries, development of cheese, right? Milk products, what you call butter and all that, right? Food industries, alcohols, production of antibiotics, medicines, or production of the proteins, enzymes, right? This all today well produced by using microorganisms with the help of genetic engineering. By transferring the gene, we are getting desirable products we are producing at the place called as industries. So what not, we have so many applications in biotechnology, in genetic engineering, that's for all the basis is nothing but what, recombinant DNA technology. So as it has so many wide applications, now as it has so many wide applications, now from the genetic engineering we developed the branch even including microorganisms, including plant and animal cell, that's nothing but bio, and it's possible to apply technology for the transfer of genetic material from one organism to other organism. So this genetic engineering, recombinant DNA technology itself emerged as one of the most important branch today. It's called biotechnology. Now what is biotechnology? So according to European Federation of Biotechnology, what is EFB here? We can say European Federation of Biotechnology. Now, according to European Federation of Biotechnology, a biotechnology can be defined as, what is defined as? A biotechnology can be defined as integration of natural science into technologies for developing desired products at industries which are useful to human welfare, which are useful to human welfare. So according to European Federation of Biotechnology, biotechnology can be defined it as integration of natural science into technologies. Why do we say integration of natural science? Because if people already know what is by combining traditional knowledge, it's combined with the modern technologies. What is traditional knowledge? It's nothing but the natural science, because ancient people already they know how microorganisms are used in conversion of milk to curd, and even in preparation of alcohol. Already people know, ancient people, that's nothing but traditional knowledge, right? That's nothing but natural science. Integration of natural science into technologies. What is this technologies? What are the technologies which we develop today? They are called modern technologies, right? That is recombinant DNA technology. Now for developing what? Desired products at industries which are useful to the human welfare, right? Or this can also be defined it as integration of technology in using cells, parts of the cells, cell constituents, tissues and molecular analogues to human welfare. Molecular analogues helpful to human welfare. Okay. So that is nothing but what is other definition you can write for biotechnology? Integration of technology in using cells, because in cell only we find nucleus, within the nucleus we find chromosome, within the chromosome we find DNA, genetic material, as parts of the cell, because nucleus is and chromosome, these all are nothing but parts of the cell. Cell constituents, meaning it contains DNA constituents, it may contain adenine, guanine, cytosine, thymine. The gene what we are cutting, it may has different sequence of constituents of the DNA, purines and pyrimidines, and tissue we are taking group of cells, and molecular analogues. Molecular analogues means what here? We are taking one organism which exactly looks similar to the other organism, so that it is easy for us to transfer the gene from one organism to other organism. Molecular analogues means we are transferring into another organism who looks similar to the other organism. So transfer of the gene from one bacteria to other bacteria, transfer of the gene from one plant to other plant, transfer of the gene from one animal to other animal. Molecular analogues helpful to human welfare. Finally, these all desired products what are produced from the cell constituents, they are should be very much essential to the human welfare. For the sake of society, for the sake of to meet the requirement of human beings, now we are integrating the technology into living organisms. That's what itself is called as what? Biotechnology. So these all are nothing but applications of biotechnology. Very, very important. So when you take the biotechnology applications, what not? Other things also you can write if you want, other things also you can write. You know that milk converting into curd, right, and even you know alcohol production, right. So even in medicines as well as correcting the gene therapy, in vitro fertilization, this all will come under what? Biotechnology applications, right. Nowadays we are producing the plants with desirable characters, plants with desired characters. Plants, they are produced with desired characters. Why? I want rice grain should be bigger. So I am transferring the gene which can give big size to the rice. right? Basmati rice, what it may be. So plant with the desired characters. So what are these? When you transfer the gene from one organism to other organism, the other organism is called as what? Genetically modified organism. Suppose you take, this is one plant cell. So this is DNA. What we are doing here in recombinant DNA technology, with the help of genetic engineering, what we are doing, recombinant DNA, we are cutting the DNA. So we are taking the fragment of this DNA, right, and this with the help of plasmid. Plasmid is acting as a vector. This is the source, right, and what we are doing, and this we are transferring into another plant cell, and this act as what? Host. So here this has different genetic material. Into this we are adding the fragment of DNA. What you call this host? The host here itself is called as what? The host here itself is called as what? Genetically modified organism. Itself the host is called as what? Itself is called the host. Now it's called as what? Genetically modified organism. Itself is called as genetically modified organism. It's called GMO. It's called as what? GMO. Why it is called GMO? From one organism we are cutting by applying what? By applying restriction endonuclease, restriction enzyme. We are cutting fragment of DNA. The DNA is carried by plasmid. Plasmid is acting as a vector, transferring the gene into some other organism. So here foreign DNA got incorporated into host organism. Now host organism has combination of its own genetic material as well as some foreign genetic material. Now this host organism produces different characteristics, and those characteristics are produced by the gene what we are transferred. That is why the host organism is called genetically modified organism. GMO we say, genetically modified organism. That means the plant can produce, now this, if it is a plant cell, now my plant can produce desired characters. What is meant by desired characters? The characters what I want to produce, I can produce. Now what you call, if this is a cell present in the plant is called genetically modified plant, because whose genes are modified. With recombinant DNA technology, with genetic engineering, the genetic original natural genetic material got added with some other new genetic material. It expresses new genetic characters which are earlier not present in the original cell. So now that organism is called genetically modified organism. That is possible by genetic engineering. It's one of the application of what is called as biotechnology. These all are nothing but what? Applications of biotechnology.